How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Keira Deneen. I'm a certified genetic counselor and your host. To celebrate February as Rare Disease Month, we're going to be learning about new research in one of the mitochondrial disorders, primary mitochondrial myopathy. And joining me for this conversation is Dr. Alejandro Dorenbaum, the Chief Medical Officer at Reneo Pharmaceuticals, who is currently studying an investigational therapy for PMM. Thank you so much for coming on the show.
Oh, my pleasure. Thank you for having me.
So I want to take people back all the way to high school. So oftentimes in high school, people remember that they learned about mitochondrial, and what they remember is it's the powerhouse of the cell. Whenever I kind of hear of mitochondrial, I still think of that. What does this exactly mean? Some people might remember that phrase, but not sure what that means in terms of what the mitochondrial do. What is the main role of mitochondrial for our cells and our body?
Well, yeah, mitochondria are really the powerhouse of the cell, and they are really, really interesting little organelles, little pieces of, that you can find within a cell, and they're unique in many respects. First, this is mind-boggling to me, but we believe that mitochondria are really not originally human organelles. They appear to have come from an infection that happened into mammalian cells, and probably because it's a very effective organelle that does the job so well, the mammalian cells decided to keep it instead of the other sources of energy that it had before. So when you look at the mitochondria, what is unique about them is that they're very tiny little organelles. There's about, between 1,000 and 2,000 of them in each cell.
Which is crazy, in each cell. Like when we look at a diagram in a textbook, there's usually like two of them. So for many years in like high school, it was like, oh, we have two mitochondria in each cell, and that is completely wrong.
Correct. They're really important because the mitochondria generates most of the energy and stores most of the energy that the cell must use to conduct all of its activities. So these mitochondria, when you look at them, they're pretty unique in their appearance as well. They have two membranes. There is an outer membrane that is fairly permeable, and then there is an inner membrane that is kind of folded with cristae. It looks like when you look at the intestines, what you see there. But that second membrane inside is where all the activities of mitochondria are happening, and what really occurs in the mitochondria is the transformation of sources of energy into energy. So the main two sources of energy for the cell are as you know, sugars and lipids, fats. So what happens is sugar goes into the cell, the glucose is converted inside the cell into pyruvate, and the pyruvate is transported into the mitochondria. Inside the mitochondria, it is converted into acetyl coenzyme A, which is a key molecule for energy in the body. This acetyl coenzyme A enters into what we all remember as the Krebs cycle, and in the Krebs cycle, it just goes around being modified by different enzymes so that it releases energy, and that energy is captured into ATP. And ATP is the molecule where we store energy that we use when we need to use energy. Lipids are similar. Interestingly, lipids enter into the cell. Now they have difficulty going from the cell into the mitochondria. So the lipids need to be transported, if they are large, into the mitochondria. So there are energy shuttles, shuttles that bring the large lipids into the mitochondria. Once inside the mitochondria, they actually break down into acetyl coenzyme A as well. They also enter the Krebs cycle, and they also yield ATP. So the main activity of the mitochondria is to generate energy and to store it into ATP. And without the mitochondria, there is simply no energy to perform daily activities. So you can imagine that, I mentioned that there are about 1,000 to 2,000 mitochondria per each cell. So cells that require more energy, like muscle cells, like nerve cells, like liver cells and kidney cells, are chock full of mitochondria. And then tissues that don't require as much energy have lesser amounts of mitochondria because they need to generate less energy. Now, what is also really, really cool about mitochondria is that they are the only organelles in the body that have their own genes.
I think that's the coolest fact about them. I mean, I geek out over mitochondria, but it's so interesting that it's different, that you have the nuclear normal DNA, as some people maybe would refer to, but then these little organelles have their own genetic information, which is one of the reasons why we think that at some point these cells were like, oh, let's take this bacteria or something and incorporate it into our own cells, as you were talking about at the beginning of the show. How is that DNA different from the nuclear normal DNA?
Oh, quite different in many respects. First, it's a circular DNA. It looks like a circle. And it codes basically for only 13 proteins. And it also codes for about 20 RNAs. Well, more than 20, but 20 ribosomal RNAs, more or less.
That's tiny compared to the nuclear DNA. I mean, that's way more.
Yeah. So, for example, in order for a mitochondria to function, you actually need all those 13 genes functioning well, but you need an additional 1,500 genes from the nucleus to be doing their job well. And from those 1,500 genes, you get proteins that form the part of the mitochondria, but also proteins that modulate the function of the mitochondria. So you could have defects in the nuclear genes that either form mitochondria or control mitochondria, or you could have defects in the mitochondrial genes that affect the mitochondria. And they behave a little differently because in the mitochondria divide, when they divide, genes get divided also, and they go into the new mitochondria. And what happens is this. All the mitochondrial genes that we have, we typically inherit from our moms, not from our dads. So the only way you inherit a defect of mitochondrial gene is through maternal inheritance. But if you have a defect, for example, and what happens is when you inherit an affected mitochondria, you may also inherit a normal mitochondria. So within the same cell, a proportion of mitochondria will be abnormal and a proportion will be normal. That's a concept known as heteroplasmy. And it's unique to the mitochondria, where you can have within the same cell normal genomes, mitochondria with normal genomes, and mitochondria with abnormal genomes. And in those cases, when there is a defect in the mitochondrial genes, it depends on the proportion of the genes that are affected, whether you will have significant manifestations of disease or not. Now, in contrast, in the nuclear genes, if you have an affected gene, all the mitochondria will have the affected problem because all of the mitochondria will be affected. You don't have heteroplasmy when there is a defect in the nuclear genes. And the nuclear defects can be inherited as in the usual Mendelian fashions, as autosomal recessive, dominant recessive, or autosomal, dominant recessive, or or as an X-linked defect as well.
It's like all the different inheritance patterns that you really could have.
You can, yeah. So that makes diagnosis of mitochondrial diseases quite difficult. But let me come back to what I was talking about in terms of the RNAs that are coded for in the mitochondria, because this is an important problem. Because there are only 20 ribosomal RNAs, when you're transcribing DNA into proteins, you're using a reduced number of RNAs. And that means that the RNAs need to accommodate for different amino acids. Typically, in the nuclear transcription, you use about 30 different RNAs, and here you only have 20. So there is more accommodation that happens, and that can lead to more defects. So the mitochondrial gene transcription is more prone to defects than the nuclear transcription. And that's important to consider in terms of these diseases.
Yeah, there's so many different factors to consider with mitochondrial conditions. You know, we've highlighted, I think, most of the common ones. PerkinElmer Genomics is a global leader in genetic testing focusing on rare diseases, inherited disorders, newborn screening, and hereditary cancer. Testing services support the full continuum of care from preconception and prenatal to neonatal, pediatric, and adult. Testing options include sequencing for targeted genes, multiple genes, the whole exome or genome, and copy number variations. Using a simple saliva or blood sample, PerkinElmer Genomics answers complex genetic questions that can proactively inform patient care and end the diagnostic odyssey for families. Learn more at PerkinElmerGenomics.com. The next generation of genomics is here. Introducing Merscope from VizGen. You can explore new dimensions through spatial context with Merscope. VizGen integrates MERFISH technology with high-resolution imaging, fluidics, image processing, and automation to deliver complete end-to-end single-cell spatial genomics solution. The Merscope platform comes with everything your lab needs to get running. VizGen built their expertise into every component of the system, so you can easily obtain and analyze high-quality MERFISH measurements. Merscope measures the expression of hundreds of genes across hundreds of thousands of cells. Its resolution ranges from the tissue scale all the way to the subcellular scale. This allows researchers to interact, analyze, and interpret the most complex data sets. Stay tuned for our interview with VizGen on DNA Today, where we explore single-cell spatial genomics. Get a sneak peek by visiting the website at vizgen.com. That's VIZGEN.com. VizGen, leaders in spatially resolved single-cell transcriptomics. Curious to take a peek inside your body's drug response? Then you should check out Picture Genetics' PGX test. Powered by clinical laboratory Fulgent Genetics, Picture's PGX test is easy to order and understand with hassle-free clinician involvement and good-looking reports. Plus, you're fully supported through live chats, emails, and genetic counseling. To order your Picture PGX test kit, use code DNA Today for 25% off and free shipping. Get actionable genetic insights today to benefit your health of tomorrow. Another thing that I'm just thinking about for mitochondrial disorders is how does this affect the body? So it's going to be affecting areas of the body that need a lot of energy, because if we have a problem with mitochondrial energy production, I'm thinking muscles, maybe eyes that require kind of more energy. Diving in specifically to primary mitochondrial myopathies, what is some of the symptoms that a person can have? Because this is a specific condition under the umbrella of mitochondrial disorders.
Yeah, so mitochondrial disorder, you're absolutely right. So the tissues that are typically most affected are the ones that have more requirement for energy. So we often see defects in the muscles, in the heart, that is a muscle also, maybe the nerves, the central nervous system that is very dependent on energy, the kidney, the liver, maybe the hearing and eyes, which are high, these are tissues that have a high requirement for energy. So yes, typically you see problems in those areas. But the overwhelming, the most common manifestation of the disease has to do with lack of endurance, with fatigue, with difficulty in performing activities of daily life, because you simply don't have enough energy to move and control your muscles well. So that is the most common symptom. And then there are other symptoms that can occur depending on how severe the mitochondrial defect is and what kind of mutation you have. But in general, patients, what I could say is patients present primarily with weakness, not either weakness or I would call it more lack of endurance. There is a difference between not having energy to perform activities or having a weak muscle. So these patients primarily have initially lack of endurance. If they have neurological complications, they can also develop weakness. Like, for instance, there is a syndrome where patients have pseudo-like strokes. And in that particular situation, these kind of strokes can cause you to have muscle weakness as well, or an ataxic walk, an unsteady walk. And often, well, there are a few syndromes where the senses are very affected. So blindness can be a manifestation of the disease. Hearing loss can be a manifestation of the disease. And generally, what I would say is that patients tend to more or less trottle along, but if they have an increased requirement for energy for any reason, that kind of tips them over the balance. And what happens is, if you catch an infection, for example, or if you are under any specific stress that requires more energy, you may not meet those demands, extra demands, and that tips you over into a really spiral of getting much sicker. Now, the manifestations of the diseases in children and adults are a little bit different. In children, the nuclear defects are more common. And in those cases, the children typically at birth, they generally, most of them do relatively well, but either within a few months or so, or even later in childhood, typically the child gets an infection and that, triggers them to be very sick for several years. And after several years, typically, they may stabilize and do better. So a child, for instance, that was able to walk initially may suddenly get sick with a viral infection. Then the mitochondrial disease manifests itself. The child may no longer be able to walk, may end up actually in a wheelchair for a while. But as that child gets older, he could potentially walk again. So we've seen that. In adults, typically, the mitochondrial gene defects are more common. And we hypothesize, we think that this is because if you have a mitochondrial gene defect, you have both normal and abnormal mitochondria. So maybe these diseases may be less severe than if all your mitochondria were affected. So you're more likely to make it well into adulthood. And what happens with adults, which I think is very interesting, is that, before they get diagnosed, these adults usually get seen by 5, 6, 7, sometimes even 8 doctors before a diagnosis is established. Now, I practice medicine and I am a specialist as well. And when children come to see me, I usually can make the diagnosis fairly quickly. And sometimes it's because I know my specialty very well, but sometimes the reality is that I get to see the patients when they are more advanced, so it's a little easier to make the diagnosis. So you can't entirely blame it on physicians being ignorant about the disease. But I think that in general, it's difficult to recognize these diseases early on in adults in particular. And because they can be very vague. Fatigue, during this pandemic, we all have experienced fatigue. We've all had days where we say, I don't feel like moving my muscles. So fatigue is a difficult symptom to pinpoint. And many patients, unless they are having one of the more characteristic findings like hearing loss or vision problems or these stroke-like episodes, then that clearly, physicians know this. I should be looking for the mitochondria. But if you don't see those things and all you see is the fatigue and the weakness and difficulty moving,
at that point,
then it's very generic and difficult to identify. But once they are identified, in the past, And in fact, when I was a student of medicine, was doing my residency training, we would biopsy everyone to look at their muscles, and you would look at the characteristics of the muscles and the mitochondria to see if there is a defect there. Nowadays, biopsies are not as necessary as they used to be because of the extent of genome testing. And with new sequencing testing, that next generation testing that exists now, and making it so much affordable, it's much easier to identify these defects.
And some whole genome sequencing I'm seeing is now including the mitochondrial genome, I suppose, if that's the right language, to include that. So I'm sure that's changing the game as well, because now we're starting to include the mitochondrial DNA as well.
Yeah, well, I'll be very, very, very humble about this. As a genetic counselor, you probably know much more than me about this.
I don't know about that.
but I'll say a few things, a few pearls that I've learned along the way. First, the problem is, you remember I mentioned there are 1,500 genes involved in mitochondrial function. Well, only about 300 genes have been identified as having defects in these diseases. So we anticipate that there will be many more new genetic defects that will be identified. So what follows from that is that there are mitochondrial panels that you can do for genetic testing, but you would only be able to see if the defect is in the known genes. But there are about 1,200 that we don't know about. So if you have a if you have a classic patient, yes, you should do targeted gene, you should do a panel of mitochondrial gene defects. And, there are panels with 20 or 30 genes you can do, and then you can corroborate your diagnosis. But if the presentation is not that classic, you're better off trying to do the more extensive tests, because you may actually identify a gene that hasn't been identified before.
Right, or if you end up having a negative, like, all right, come back a few years later, maybe now we've figured out one of those genes now. It's been added to a panel. Now we understand that's not a VUS anymore, variant of unknown significance. This is actually mutation, a pathogenic variant. We say now we understand this is playing a role with the mitochondria. So that's kind of the case with any kind of genetic testing. Like, there's always updates.
Yeah, and interestingly, I mean, you touch on a very important point, which is often you see very changes that are of unknown significance. So sometimes you actually have to then biopsy these patients, take fibroblasts, grow them in the laboratory, evaluate the mitochondrial function, and see, okay, does that genetic variant correlate with a defect in the mitochondrial function? And the main test that you would do in the mitochondria would be to look at the ability of the mitochondria to do oxidative phosphorylation, which is the process, this Krebs cycle. You evaluate whether they can generate energy properly. That's the way to identify new variants.
Did you know 90% of people carry at least one drug-reactive gene variant? Yes, it's actually that common. That's why Picture Genetics PGX test is so helpful. The at-home test kit helps you personalize your medication planning based on your specific genetics. The test is powered by Fulgent Genetics, a clinical laboratory that uses pharmacogenetics to analyze your genes to help you identify what meds work best for you. No more blind guesses. PGX testing gives you genetic insight on your body's ability to process different drugs. Tests come with a clinical report and genetic counseling, so you're fully supported and educated about how the results could impact you. Use the power of genetics to find the right medications with Picture Genetics PGX test. Visit picturegenetics.com and use code DNA Today for 25% off and free shipping. Again, that's picturegenetics.com with code DNA Today for your Picture PGX test kit. Get actual genetic insights today to benefit your health of tomorrow. Oh, and stay tuned for a full episode about pharmacogenomics with Picture Genetics coming soon. VizGen is dedicated to pioneering the next generation of genomics, providing tools that demonstrate the possibilities of in situ single-cell spatial genomics. These tools are enabling researchers to gain insight into the biological systems that govern human health and disease. Stay tuned for our full episode with VizGen, where we explore single-cell spatial genomics. Get a sneak peek by visiting their website at vizgen.com. That's VIZGEN.com. VizGen, leaders in spatially resolved single-cell transcriptomics. Did you know PerkinElmer Genomics was one of the first laboratories to offer whole genome sequencing on a clinical basis? Whole genome sequencing can maximize clinical diagnostic yield for patients. With a turnaround time of 6 weeks, PerkinElmer's whole genome sequencing test is designed to provide access to additional valuable information compared to an exome. PerkinElmer Genomics provides one of the world's most comprehensive programs for detecting clinically significant genomic changes. PerkinElmer Genomics delivers knowledge that can empower health. PerkinElmer is a global leader committed to innovating for a healthier world. Join their mission at PerkinElmerGenomics.com. And also, stay tuned for our interview with PerkinElmer, where we're going to be exploring the power of whole genome sequencing. I'll also tease that my guest for this episode is a world-renowned geneticist, so you are not going to want to miss this one. But while you wait for the episode, head over to PerkinElmerGenomics.com to start discovering all PerkinElmer has to offer. So we've been talking a lot about mitochondrial disorders kind of in general, of the different symptoms we can have and the genetics behind mitochondria. What about for primary mitochondrial myopathies? Are these also the symptoms for this disorder? Like, is it more particular than in general for mitochondrial disorders, or is it also very generic?
No. Well, here's the interesting thing. So mitochondria, as I said earlier, they're prone to have defects, and as we age, the mitochondria mutate. The DNA in the mitochondria mutates. And experts postulate that these increases in mutations in the mitochondria are part of the aging process of the cell, and the cells become less efficient, and that's why we age, in part. Not completely, but in part. So if you take the cells of an elderly person, you will find a proportion of mitochondria that are mutated, always. And the question is, how much is that affecting this individual? So we refer to primary mitochondrial myopathies as the diseases where there is a primary defect in the genes that either code for mitochondrial proteins or code for proteins that control the mitochondrial function. But there are many other diseases where the mitochondria are affected. For instance, there is evidence that in Parkinson's disease, the mitochondria are affected. If you look at chronic fatigue, for instance, there is evidence that the mitochondria may be affected. In Alzheimer's, the mitochondria are affected. In ALS, the mitochondria are. There are many diseases where the mitochondria are affected. So we subset the diseases where there is a genetic defect specific to the functioning or structure of the mitochondria as the primary mitochondrial diseases, and they manifest in the form that we discussed before. Now, we are particularly interested in what we call primary mitochondrial myopathy. And primary mitochondrial myopathy means a defect in the muscle, myopathy, that is caused by a defect in the mitochondria. So, you can have defects in your ability to move your muscles that are not related to the mitochondria, like if you had a stroke or you have a bone defect that doesn't allow you to walk well, or you have muscle wasting because you've had a cast put on your leg for a long time or whatever. So there are many defects in the muscle activity. But when that muscle activity is affected because the mitochondria are abnormal, we call that primary mitochondrial myopathy. And that's what we are studying at Reneo, the patients with primary mitochondrial myopathy.
So what is Reneo doing to help diagnose more patients through genetic testing programs? Because as you said, it can be hard to diagnose.
So there are societies for patients with mitochondrial myopathies. As you know, the patient advocates and groups get together, and we're working with these groups to facilitate genetic testing, because we think that if you, there are two barriers to getting or I would say more than two, but at least two barriers that we can address to proper diagnosis of these diseases. One is education of the clinicians, and they need, because these diseases often manifest in a very non-specific way. You want to have a high index of suspicion, so you want to educate clinicians that they should suspect these diseases. But once they suspect them, if you can make genetic testing affordable and easy and reachable to the patient, then it's much easier for the physician or the genetic counselor to propose doing the testing. So we're actually doing a program to fund genetic testing for patients who present with symptoms suggestive of a primary mitochondrial myopathy, who may not have access because of insurance problems to the genetic testing.
And so as part of this program, Reneo is working on a treatment for people with primary mitochondrial myopathy.
Well, so we are evaluating a drug to treat patients with primary mitochondrial myopathy. Yes. And what is interesting about this drug is that this is a drug, so as you know, activation of genes is highly regulated in the cell. And there are receptors in the nucleus that kind of tell the nucleus, I need this set of genes activated because I'm going to do these kind of activities. So we all know, I mean, the classic nuclear receptor is the receptor for the steroids. When you give steroids to a person, the steroids go into the cell, they activate a nuclear receptor, and that nuclear receptor activates multiple genes, and you have all the changes that happen when you take steroids. So in that fashion, there are a set of other nuclear receptors that are involved in energy metabolism, and they're called PPARs, PPAR. So these PPAR receptors, they come in three flavors. There is alpha, delta, and gamma. And there are drugs to activate PPAR alpha and PPAR gamma. And these are drugs that are used to treat patients with high lipids in the blood or patients with diabetes, because when you activate PPAR, these are PPAR agonists, you activate genes involved in metabolism. Some of those genes, like when you activate PPAR alpha or gamma, activate the genes that are involved in processing sugar and lower the sugar, or processing lipids and can lower the lipids. So that's why these drugs are currently in the market. There are no drugs approved for PPAR delta. And PPAR delta is a little different than alpha and gamma in that alpha and gamma are primarily really intrinsically metabolic activators, and they're more present in the liver and a little bit in the kidney, whereas PPAR delta is present in almost all tissues, primarily in the muscles. So that's why we were interested in this PPAR. When you activate PPAR delta, you activate genes in the muscles that are involved in oxidative phosphorylation, in improving
that's super important
mitochondrial
for
function.
Disorder. Yeah, that makes sense.
And they also activate genes that generate more reproduction of more mitochondria in your cell. So you can increase the number of mitochondria and you can improve the function of the mitochondria. And in fact, we tested our medicine, our medicine candidate, in cells of patients with, for instance, fatty acid disorders. So these are patients who are missing some of the enzymes to metabolize the fats in the mitochondria. And what we were able to show is that we can increase the production of energy in those defective cells. So we know we can improve the function of the mitochondria that are affected.
Wow, it's really cool just what you're able to look at, and the mitochondrial is just so interesting. If people want to find out more about the study, which is called STRIDE, if people want to find out more about the study, where can they go?
We have for STRIDE a website. When you click that, it directs you to a website that will ask you a few questions and give you some information as to whether you might be a good candidate or not for the study.
I think it's pmmstudy.com.
That is
sounds
correct.
right? Okay, yeah. I'm on it now. Looks good, yeah.
Yeah, pmmstudy.com. But also, I always advise people who are interested in learning about this to going to clinicaltrials.gov and just you can enter either Reneo or the name of our drug, which is REN, REN001, or you can enter PMM clinical trials, which is primary mitochondrial myopathy. And you know, not only our study, but other studies that are being done by other companies will appear there, and you can look at ours and you can look at the studies that others are doing as well, and kind of figure out what works best for you. You know, in my career, I have most of my life dealt with diseases for which there is no clear treatment or cure. I have had an interest in developing medicines for difficult to treat or rare diseases. And I believe that when you're dealing with diseases like this, treatment for the patient is very important. But if you can do that treatment in the setting of research, is even more valuable, because we don't have a good treatment and we don't have a good cure for these diseases. So I really encourage, you know, both clinicians and that are treating these patients and patients to consider participating in clinical trials, because hopefully that will help figure out an effective treatment and eventually, hopefully, a cure.
I think that's very well said, Dr. Dorenbaum, and there's just so much out there to learn, and I think if patients can be a partner in research, it goes a long way. It can just help so many people, as you said, of having research be involved in these development of treatments and everything. Mitochondrial disorders and just the genetics is very fascinating. So I encourage people to just learn more about it. You know, we've covered quite a bit on this episode, but just really want to thank you, Dr. Dorenbaum, for coming on the show and geeking out with me a little bit about mitochondria today. For more information about today's episode, visit DNApodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@DNApodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Keir Deneen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA.